Our next
Thanks.
speaker is Sarah Jones. Yep, and she is a fourth year PhD student at Florida State University. And she'll be discussing transgenerational transmission of anxiety and working memory deficit following paternal aspartame exposure. Sarah?
All right, thank you, Tia. I'm going to talk to you today about heritable behavioral phenotypes following paternal aspartame exposure. So some of you might be familiar with aspartame. It's an artificial sweetener that can be found in over 6,000 food products. Aspartame sweetened products are appealing low-calorie alternatives to many of our favorite snacks and junk food items. Some products containing aspartame include sugar-free gelatin, condiments like sugar-free ketchup, and the majority of diet sodas, including the Diet Coke pictured here. So what's known about aspartame's effects on the brain? Well, aspartame has been shown in a handful of studies to potentially cause anxiety, memory impairment, depression, and other behavioral or neurological consequences in animal models and in human research. But aspartame is considered safe according to the FDA. Therefore, I became interested in a mouse model to know how aspartame exposure could impact behavior. So to study the effects of aspartame, I developed an exposure mouse model. I delivered aspartame in drinking water because consumption is oral and people are consuming it through what they eat and drink. I used C57BL6 male mice. One big question was how much aspartame to provide. And I referred to the acceptable daily intake determined by the FDA, which is 50 milligrams per kilogram of body weight. So using an established conversion formula that takes into account differences in pharmacokinetics and body surface area, I calculated that for a mouse, a 0.015% aspartame solution would provide approximately 50% of the human acceptable daily intake. So to put that into perspective for you all, that's equivalent to drinking about 6 cans of diet soda per day. So I wanted to examine behavioral consequences of aspartame exposure in this model. So I used behavioral assays to measure anxiety, spatial working memory, and depression. So now I can share with you my data starting with anxiety-like behavior. So to assess anxiety, I used an open field test. The way this behavior works is that a mouse is placed into the box and it's allowed to freely explore. And so the time the mouse spends in the center of the box and the periphery of the box is recorded. Normally, mice spend about 30 to 40% of the total time in the center, but a mouse with an anxiety phenotype may spend reduced time in the center. So this graph is showing the time in seconds. So I ran a t-test to compare the two groups and found that the aspartame exposed mice spent significantly less time in the center of the open field. So seeing that male mice consuming aspartame at 50% of the acceptable daily intake are displaying an anxiety-like phenotype. The next behavior I wanted to look at was spatial working memory, and I used a Y maze for this. For anyone unfamiliar, it's shaped like a Y and it has 3 equally sized arms, and each arm has a visual cue at the end of it. Normally, mice will not re-enter an arm they just explored and will instead go into a new arm. A spontaneous alternation is scored for each 3 set of choices where no repeated arm entries occurred. So for example, a mouse could move from arm A to arm B to arm C. An incorrect alternation would be something like moving from arm A to arm B and then back into arm A again. We calculate percent spontaneous alternations using the number of correct alternations and the total number of entries made. Normally, a mouse has an index of about 65%. So here's the data for the spontaneous alternations. I ran a t-test to compare the two groups and saw that the aspartame group displayed significantly reduced spontaneous alternations, which may suggest a spatial working memory deficit. The last behavior I used is the tail suspension test, which is used as a measure of learned helplessness and depression-like behavior. The way this test works is that when mice are suspended by the tail, they instinctively will try to escape and move their limbs. Immobility is defined by a lack of movement in all 4 limbs, and you can measure the amount of time the mouse spends immobile. Mice that spend increased time immobile may express a depression-like phenotype. This graph is showing the time immobile in seconds, and a t-test showed no significant difference between the aspartame and the control group, suggesting that aspartame did not impact depression-like behavior in my model. So what I found was that male mice receiving aspartame at 50% of the acceptable daily intake displayed significant anxiety-like behavior and spatial working memory deficit, and I saw no change in depression-like behavior. So the big question I then had was, could these aspartame-induced phenotypes be heritable? So to study this, I exposed male mice to aspartame in their drinking water for 12 weeks using the exposure paradigm I just mentioned, at which point I bred them with drug-naive female mice that have never been exposed to aspartame to then make the F1 generation. At postnatal day 60, I then ran the F1 mice through those same behavioral assays to assess anxiety, spatial working memory, and depression. So here's the results from the open field, which measures anxiety-like behavior. In white are the F1 mice produced from fathers consuming plain drinking water as a control, and in gray are the F1 offspring produced from fathers exposed to aspartame for 12 weeks. So when I ran a two-way ANOVA, I saw a main effect of paternal aspartame treatment. So the F1 aspartame mice displayed significantly reduced time in the center of the open field, which suggests anxiety-like behavior. I next ran the Y maze to assess spatial working memory. And a two-way ANOVA showed a main effect of paternal aspartame treatment again. So the aspartame F1s are displaying significantly reduced percent spontaneous alternations, which suggests a working memory deficit. I then used the tail suspension test to assess depression-like behavior, and I saw no difference in the time immobile between water and aspartame F1 mice. So coming back to my question of, are aspartame-induced phenotypes heritable, my data shows that yes, they can be. This is a really exciting finding because aspartame is something that people are widely consuming today, and that could impact future generations. So this brings me to my next question, which was, well, when during the father's exposure period does this anxiety-like behavior emerge? Is it present after 1 week of aspartame, or does it take those full 12 weeks? To answer this question, I exposed a cohort of male mice to aspartame and ran the open field test every 2 weeks. I found that at 8 weeks of exposure, male mice consuming aspartame began to show that anxiety phenotype, which is also present at 10 weeks and 12 weeks. We were next curious what would happen if I took those males exposed for just 8 weeks when the phenotype first emerges and bred them to produce F1s. I used the same breeding paradigm as before and then ran the F1 mice through those 3 behaviors. So here's the data from the open field. What I saw this time was no significant effect of paternal aspartame treatment on time spent in the center. So the anxiety phenotype that was present at 12-week in the 12-week aspartame F1s was not present in the 8-week aspartame F1s. When I ran the Y maze, I also saw no significant effect of paternal aspartame. So again, the phenotype that was present in the 12-week F1s was not present in the 8-week F1s. I then ran the tail suspension test, which showed no significant effect of paternal aspartame exposure. This is comparable to what I saw with the 12-week F1s. So to tie it all together, what I've shown to you today is that male mice consuming aspartame at 50% of the acceptable daily intake showed significant anxiety-like behavior and spatial working memory deficit. Offspring produced following 12 weeks of this paternal aspartame exposure also display anxiety-like behavior and spatial working memory deficit. The anxiety-like phenotype in the father emerges at 8 weeks of exposure, but offspring produced following 8 weeks of paternal aspartame exposure do not display behavioral alternations. Ultimately, this suggests that in this model, for these behavioral phenotypes, I'm seeing that the somatic phenotype in the F0 father may be appearing before the heritable phenotype. And with that, I want to thank Florida State University and the College of Medicine, as well as Dr. Bide and his lab, where I'm currently pursuing my PhD. I also want to thank the Beyond Genes Conference for providing me with this opportunity to share my work. And of course, thank you to all of you for listening. And with that, I'll take any questions you may have.
Thank you so much. Great presentation. Again, the Q&A box is open. I have a question. So after you're done looking at these behaviors in the mice, are you looking at brain tissue methylation?
Yeah, that's something I'm actually really interested in looking at. I've collected tissue samples from brain regions like the prefrontal cortex, amygdala, hippocampus and hypothalamus in
Hmm.
those F1 mice. So I have those samples, and so I'm really interested in looking at methylation in those brain regions, especially based on the behavioral phenotypes that I've seen.
Yeah. Are you also going to look at the control 8-week and 12-week F1s as well, or just the 12 in control?
We're really interested in looking at the 8 weeks as well, just because
Hmm.
I don't have the behavioral phenotype there.
Yeah. Yeah, I was going to say, okay, so it looks like we have a few questions. This is from Christopher Falk. So again, we see a necessary 12-week paternal exposure, but why? Why does the F1 phenotype not occur until 12 weeks of paternal exposure, while 8 is not enough?
Yeah, these are both really good questions. I started with 12 weeks because we have, as Deirdre, who talked previously, kind of mentioned, 12 weeks for some reason is kind of the sweet spot to start to see the behavioral the behavioral phenotypes. We're not really sure. It may have something to do with the sperm cycle. But truthfully, at this point, it's really hard to tell. And I'm hopeful that by actually diving into the molecular mechanism and comparing the brains at 8 weeks and 12 weeks, and also the sperm, that maybe I will have a better answer of why it takes 12 weeks.
All right, perfect. And then I had one last question. It was for the F0 male behavior tests. When you did the behavior test, how long were the males exposed to the aspartame in their water?
Yeah, that's a good question. So the open field is 12 weeks. The Y maze is 13 weeks.